Great.
Okay, I need, if someone, the host has to enable me to share the screen.
Ja, och sen får du klicka i så jag kan pausa här, för det kan inte jag göra än, Mattias.
And now I can. Good. Okay, guys, do you see my screen?
Yes.
Okay, good.
Yeah.
All right. Yes. Okay. Well, before I start off this talk, if you guys, you have any questions from yesterday presentation, please just let me know now, or just feel free, or after we finish this presentation. Okay. Well, so we're gonna talk in the next couple of hours about two, as you can see, about esters and polyphenols. So basically, we're gonna just specifically gonna be, pretty much biochemistry and chemistry. So I will try and explain it, the easiest way possible. But I know it's a complex topic, I know chemistry is not so easy to understand. So please you can stop me anytime if you don't follow me. Okay. So we start first with esters. So why we are talking today about esters? Because well, in the previous course, people were, many of the brewer students, they were interested in esters because in the literature and in general, when people talk about beer, they're talking about estery beers and so on, but people don't know exactly what esters mean, so what type of the nature of these compounds. So so what you can see here on the slide, esters are the most important flavor active compounds you find in beer, and they are pretty much fermentation byproducts, and they are derived by, as you can read, from an alcohol, so mostly higher alcohols, and unsaturated fatty acids. Fatty acids, they are derived, they're from lipids, and unsaturated means that it's about the bond between the carbons that form the fatty acids. So as you hear, very common expression of unsaturated oils, or for cooking, or saturated ones. So basically the unsaturated ones, they have a double bond between the carbons in these long chains of carbons which form these compounds. And these double bonds are easy to break energetic wise by the cells. Can be not exclusively brewing yeast cells, but also human cells or any kind of living cells. So which and that's why they say, in general, that unsaturated fats, they are healthier, with quotation marks, than the saturated ones, because they are much easier to break by the organisms. So and in the case of the human beings, so the human cells is more to break down saturated fats is much harder. So then so normally what they clog, veins and everything else. But basically that's the principle. Now, when it comes now to the esters, they are around 90. They found around 90 esters, but it's really a massive list of compounds we're going to show you soon. And but the important thing I want you guys keep in mind is that esters normally are perceived from the nostrils, so from our olfactory sense, not from so we cannot taste them, but we can smell them. So and also obviously that's the relevance of these compounds in beer. And another thing is that we're going to show in the next slides, the esters, because they're derived by alcohols, higher alcohols, so it's very dependent on the concentration of higher alcohols present in the beer. And the main compounds that they normally quantify in beer, and the main esters are the ethyl acetate and the ethyl hexanoate, and both they give you more solvent. The other one more like apple kind of notes, which certain concentration, like everything, chemistry, and there it's not like there are limits, and there you can. It's not the more the better. There is a point that it could be a positive impact in the beer, and when you exceed that, it goes negative. And the other one, which you're gonna see, which is very popular, that is in the literature, is the azomale acetate, which give you these banana notes for mainly in this kind of beers for like German wheat beers, Hefeweizen, Weissbier. And the threshold of this particular ester or the azomale acetate is much lower in comparison to the rest of the esters present in beer. So the threshold range of the esters in beer, they are between 15 to 20 milligrams per liter. And of course in ales, which ferment at higher temperature, so the production is also is higher than lagers. They can reach between 30 to 40 milligrams. The factors to promote the esters, there are many. There is not just one. So that's the main reason of this talk. But something important is that the esters are not formed at the same rate as the higher alcohols. So it's important first that normally the formation of esters takes place at later stages of the fermentation in relation to the formation of higher alcohols, which are produced pretty much at the same time as the ethanol. And so another thing that we actually yesterday, we were talking about is just the reason of aeration in wort to promote the formation of the cell membrane, because the yeast requires to build the membrane with the synthesis of esterols, so which are required, which require the acetyl CoA, which is one of the most important intermediate compounds for the regulation of the brewing yeast metabolism. And it's very important because regulates for many cycles, but one of the main ones is the Krebs cycle. And the Krebs cycle is what the yeast cell use for convert to basically produce energy from the substrate, just to and when we say substrate, from the glucose and one of the fermentable sugars into ATP. So when you has a higher levels of aeration, and then you're promoting more the formation of the esterols, therefore you will have less acetyl CoA available for the production of esters. Okay. Here is I don't ask you to learn this by heart, because I think after a couple of hours of this talk you might forget many of these names, and the point is not to learn by heart, but just to understand the principle, how they form. And so as you can see here in this diagram, representing the yeast cell, the formation of esters are related, as I mentioned earlier, between alcohols, mainly the higher alcohols, and the fatty acid CoA, which this one will precursor of the product of the formation of fatty acids. So that's the most important you need to keep in mind, when it comes to esters. The relation between higher alcohols and fatty acids. Okay. Here is just an example of how to form probably the most or the major ester present in beer, which is the ethyl acetate. So from the glycolysis metabolic pathway, it's gonna result to the formation of pyruvic acid, and through the for the fermentative process, there will be a decarboxylation to produce the ethanol, which is an aldehyde. And that one will result, the reduced form will produce to the ethanol, which is our, as we know, is the main, is the alcohol, as we call, the main alcohol present in beer. And from the other side, from the same pyruvic acid is going to be derived to the acetyl-CoA, which is going to regulate, as I mentioned, the Krebs cycle, but also can regulate the formation of, of esters. And this is a clear example. Here, I took this massive table. Actually, is not for beer, but for wine. I took it from one of the AAB presentations. As the AAB is has been running this company since the 60s in the wine industry, and beer in the last 15 years, as far as I understand. But I wanted to show you with you guys, the massive spectrum of flavor tones, flavor connotations, aroma connotations that you can get from the esters, and depending on the size of the molecule, so you can associate here, what is the more predominant, area that you want to express. If you want to express more, citruses or tropical aromas or more coconuts, and in which range you're dealing with. But that is just it's a very informative table, I think. Here's what I was mentioned earlier. So how the relation between the formation of higher alcohols and the formation of esters. Okay. Also, you can relate it to the normal concentration range found in beer. So as you could see in this, in the curve on the left-hand side, you see that the production of higher alcohols in beer, you have in a range between 70 to 90 milligrams per liter, while esters, you have even, a quarter of them, not even a half, between 12 to 20 milligrams per liter. And also you can see in the fermentation diagram that how the formation of these two compounds are formed, during, in relation to the yeast growth. So as you can see, when it reach, the lag phase, is kind of both of them, forming in a linear form, but the esters keep forming in a later stage of the fermentation, even when the concentration of the yeast declined, and it's already in a stationary phase, or even in a dead phase, even, so but in a stationary phase, I would say. So yeah, that's important that you can compare, to always keep in mind the relation between the esters and the higher alcohols, and knowing this, also you can trick from the practical brewing point of view, how you can promote more esters, or and in relation to the formation of higher alcohols. Here, just wanted to show you one of the typical analysis of flavor active compounds in beer for fresh beer, because here it doesn't appear the beer engine compounds, but on the left hand side you can see the relation between, well, the most important esters, which is the ethyl acetate. Sorry, you would see it now in German. I took this table from the VLV. I don't used to, for a strange reason, to translate to English properly, but anyway, it's you got the ethyl acetate, the isoamyl acetate, and the 2-phenyl ethyl acetate. These three, they are like the most important ones, I would say. And as I mentioned earlier, the ethyl acetate will give you more like a fruity kind of solvent kind of aroma, while the isoamyl acetate will give you more kind of banana notes. which is the one that you want to promote when you're brewing German beer, with beers. And the 2-phenyl ethyl acetate give you more floral ones. So actually that's kind of positive to find this kind of ester in lagers, for instance. And of course, if you want to for IPAs, but floral is not the most common attribute that you're looking for. IPAs more citrusy and tropical notes. And on the other hand, you can compare, the most important higher alcohols, which is, 1-propanol, 1-isobutanol, 2-methyl-1-butanol, and 3-methyl-1-butanol. And actually, the breakdown, the byproducts of the oxidation byproducts, when these compounds or these higher alcohols get oxidized, they normally they form the aldehyde form, which is the 2-methyl-1 aldehyde, which is one of the main beer aging compounds found in beer. So I just wanted to show you here the comparison between the concentrations between the higher alcohols and the esters, and in beer. And we are talking about, in this particular case, just a Pilsner, a regular Pilsner. Okay. And on the right-hand side, the table on the right-hand side, took it from the George Fix. It's an old book from the 1999, but very good book. Unfortunately, George Fix is not with us anymore. It's an interesting table, how he's comparing the impact that you have in terms of the fermentation temperature. So in the concentration of the higher alcohol, the most important higher alcohol, and the most important ester. And as you can see, there is an impact, a clear impact of the fermentation temperature. So the higher the temperature, the fermentation temperature, the more you promote the formation of esters. And it's not the same case for higher alcohols. So as you can see, between 19 degrees to 24 degrees Celsius, there is not really an impact on the formation of higher alcohols, at least not for 1-propanol, which is the main one, which is a reference, So that's interesting, And of course, also tells you that the more you increase the fermentation temperature, that means that there is more the reactions of the higher alcohols with the fatty acids, and specifically the fatty acid CoA, in order to form more esters. Okay.
Wait, I don't quite understand that last. So the more alcohols you produce, the higher the temperature. It didn't look like there was very much of a difference.
No. Yeah, exactly. Here's just the impact that there is pretty much a point I want to make is that there is no impact on the temperature and the formation of the higher alcohols. Yeah, but here, I cannot. You're right that you pointed out, but it's clear that also there is a major formation of esters, and those esters then need to be formed from the higher alcohols. Okay.
That just goes that seems counterintuitive. They always talk about the formation of higher alcohols at higher fermentation temperatures, though.
Yeah, no, it's not the case. I mean, yes, as you can see, obviously there will be a higher formation of higher alcohols at 19 degrees, because here we're talking about ales, right? You will have much lower in terms of the lager. But the point is that it wouldn't be linear. This is the point I want to make, or also this table wants to make. It's not like the more you increase the temperature, the more higher costs you're going to produce. So here, at least in this particular table, tells you that there is no a big variation in terms of 1-propanol in comparison to the formation of esters. I don't know if I'm explaining myself.
Okay. Sorry. Was propanol a higher alcohol?
Yes.
Okay. All right. Thank you.
Yeah. So you can see here in the table on the left-hand side you know, propanol is also considered as one of the higher alcohols, which are detected and quantified for as a flavor active compound in beer. Yeah.
Okay.
Now, if we move to the next slide, here I just wanted to show you the. There is some. Here there is one also common ester called ethyl hexanoate, and there is another ones that they're not all of esters provides a positive impact in beer. There are some others that, like as you can see on the table on the bottom, that for example, ethyl hexanoate gives you more soapy kind of estery notes. So and the same goes with the ethyl acetate. Even is one of the main ones, or the one which presents a major concentration in beer, as you can see. It gives you, depends on the concentration, can give you like a fruity, pleasant aromas, but can go even to, like a solvent kind of aroma as well, which is the same case, well, actually in general, all the flavor active compounds, fermentation by flavor active compounds, they like higher alcohols, they can be pleasant, they can give you fruity notes, but if you go too high, as we know, that you can get this kind of solvent, even sometimes slightly plastic, and cloying kind of notes. So that's important when I was talking yesterday, that you should not exceed the, fermentation temperatures, at least what I learned, in my case, not going, above, me between 19 to 21 degrees is enough. But if you go above that, 23, 24, 25, you're really accelerating the formation of these compounds, so you need just to be careful about it. But again, this is just, there is no black and white. It depends a lot on the matrix of your beer, also the levels of your hops, it's the whole harmony of all the flavor components that you have in your beer. But at least what is clear is that if you have a higher temperatures, you're gonna pretty much promote all these flavor active compounds. And I will tell you more, some other factors important in relation to esters. Here, just wanted to show you the impact that has the temperature and the pressure, and the aeration, and all these factors that they really have an impact on the formation of esters. So one is the temperature that we already saw in the previous slides. And the second one is, when you ferment with a, let's say, pressure fermentation that you can see on the left-hand side, how is if you has a higher internal pressure in the tank, you're going to repress the yeast, and that will lead to less formation of esters. As you can see here, there is a quite informative table at, with different temperatures. So, or it's going to slow down, the formation of esters. So this is one thing. The other that is not considered here, I mean, it's not, it's something I mentioned earlier, is the configuration of your fermentation tank. If you have like if the hydrostatic pressure is the higher the hydrostatic pressure, the less formation of esters you're going to get. And that's why this philosophy I mentioned earlier from Heineken, that they when they to produce their beers, when you want to get a license to produce their beers, you don't produce Heineken beer on CCTs, and you can add this to Spendrups. And then another factor is the pitching rates. You remember yesterday we were talking about that is known that the dry active yeast, they produce less esters than the liquid ones. I mentioned earlier, for a couple of factors. First for mutation reasons, but also the main one is also because of the concentration of yeast cells. And as you can see here, there is the message is here that overpitching or major concentration of cells is going to reduce the formation of esters. And the same goes with aeration, because of the previous point I made. You're going to favor the formation of the esterols, and you will have less acetyl-CoA for the formation of esters. And when it comes to the OG, the extract, so it's clear, as you can see, well, the higher the extract, the higher the level of esters, and the fermentation temperature that we're just corroborating from the previous slide. So here's another factors, it's just, again, kind of sort of reaffirming the previous slides. Also the presence of glucose, in wort, also will promote the levels of esters, the impact of formation of esters. The intensity of the fermentation as well, with the temperature. Here is kind of strange. I took this one from the VLB because there is mentioned that the more the unsaturated fatty acids, the less the impact of esters. That doesn't make sense to me, because you need them for the formation of esters. The other one, well, the pitching rate we already mentioned earlier in the previous slide. Obviously, this is super important, is the G strain. There are some strains that they produce higher levels of esters than others, no matter the fermentation conditions. And the wort aeration, when it comes to the wort aeration, you will find many inconsistencies in the literature. Some literature, they mention that by increasing the aeration of your wort, you will have, major formation of esters. But as you can see in this table by the VLB, it's like a plus minus. So we are talking about, theory, right? We're talking about chemistry, theoretical chemistry, and and biochemistry. So as I mentioned earlier, so the more you increase the aeration, the more you're promoting the formation of esterols, which requires the acetyl-CoA. So therefore, less acetyl-CoA available for the formation of esters. In short, in the chemistry tells you that the more you aerate the wort, the less formation of esters. But this is theoretical chemistry. The same goes with the pressure. Well, the pressure is obvious, that also will repress the yeast, as I mentioned earlier. Now the kreuzening, which is a method I mentioned earlier as well, that one also will promote, because you have a high activity of the yeast in the kreuzen, so also will promote the formation of esters. Also, it's a common practice. The Bavarians, if you go to small breweries in Bavaria to produce wheat beers, they have a common practice to do this kreuzening to promote the aromas of banana in their wheat beers, using open fermentation vessels. What else? The configuration of the tank was related to the hydrostatic pressure. The hazy of the lautering, that one, I don't know why they mentioned this, because the hazy, as we know, the haze that is related, when we talk about haze, everybody's thinking about the polyphenols and proteins haze, which is the most common, the most known type of haze, the protein polyphenol haze complex. But also there is another type of haze. There are the beta-glucan haze, which also is from brewers, they trying to promote when they brew, hazy IPAs, which is a carbohydrate haze. And also there is the chemical haze, which is induced by the formation of, like the beer stones, which basically is calcium oxalate. And there is the biological haze, which is promoted by yeast, by non-flocculant yeast, or even can be by contamination. So in this case of the hazy lautering, it's not clear to me. but the presence of zinc, yes, because the zinc is a growth cofactor, which is needed actually in the formation of the acetyl CoA, for example. So the zinc plays even as a tiny role, but is essential in two important metabolic pathways in the brewing yeast, which is the formation of the glycolysis, which breaks the glucose into pyruvic acid, and then from there, and also from there goes to the Krebs cycle. So in both cycles, the zinc is present. So that's why it's important, and obviously, with this, the message on here is that the message is to the brewers to consider also the addition of nutrients, which they are enriched in zinc. Yeah. Well, the last point, multiple fermentation tanks, also called Drauflassen, the Germans, that this is basically is when you are, filling up your fermentation tanks sequentially, and normally what you do is you heavily aerate the first batch, and then gradually you are reducing the aeration. In some cases, you don't even aerate aerate the wort anymore, because the yeast already reached the log phase, but here the point they want to make is that if you are using this method, you might reduce the formation of esters. Yeah, well, this is about esters. I don't know if you guys have any questions.
Yeah, just a fast one.
Yes.
You talked about ethyl acetate being fruity, isoamyl acetate being banana, and then you were talking about a third that was flowery. Which one was that one?
I was trying to find the slide. Which one? These ones, you mean?
Yeah.
The ethyl acetate, yeah, give you more like fruity aroma. If you exceed, if you go really higher concentrations, and they get like a solvent kind of,
So is it the
ethyl acetate? I can give you the correct names. Should be.
No, but so ethyl acetate was fruity, isoamyl acetate, that was banana, but the one that you said was flowery.
The flowery is the 2-phenyl ethyl acetate. I can point you out.
Okay, yeah, I see it.
This one. This one is this definitely give you these floral notes, and I will show you more tomorrow when about the nutrients, because this is something I'm currently working in a research project and product development for AAV, and this particular compound is of great interest for this. For if you want to explore floral notes in your beer, you want to get this one together with some thiols that I mentioned earlier. That also give you some floral notes, and some esters and some mercaptanes, which is also tiny little compounds. But this one is well known, that give you floral notes. Yeah.
Awesome.
Yeah. Any other question? No. Okay, so then we move to the polyphenols. This is another topic that is of major interest for brewers, at least when we heard some comments last year. And actually the polyphenols, that were not really in the literature. You will not find much after, I would say, until the early 2000s, and because there are people in the brewing science, they already knew about the relevance with in terms of the complex with polyphenols and proteins, but also the polyphenols it's a very important compounds, because they have different functions in the brewing process. So and they're found in higher levels actually in nature. So so what are polyphenols? They're phenol rings, you know, that mostly that you find it in the barley husk or in the cereal husk, and also you can find it in the aleurone layers, you know, just beneath the husk. And they are also in a big concentration in the hops, you know, in the hops matter. So they're around 0.3% in the barley dry matter. So you would say that's not much, but they play a very important role in the brewing process. And the reason why they exist in nature, at least what we know, is that it's like a protective mechanism for the cereals and the plants. You know, you find higher levels in the trees, you know, and the leaves. Is against the UV radiation, and also and I would say mainly against the pathogens, you know. So because the polyphenols are toxic to insects and fungus, and also well, in general, also human cells, you know. So we are not able to consume, you know, higher concentrations of, you know, polyphenols in our food, and also that's why our body, our taste buds, give you the sign when you're eating something with high levels of polyphenols that used to be very astringent and bitter. So that the bodies give you the sign to say that, like, don't overdo with this, because otherwise you're going to poison yourself. So so this is, well, the functions that has the polyphenols in nature. And as you can see, also in the hops, it's a higher levels than the barley. And this is something I really want to point out, because you find more in aroma hops than in bitter hops, you know, and also because this is, well, because of the variety reasons, but also because also we use always higher levels of aroma hops or concentrations than in bitter hops. But one thing I really want to point out is, you see the relevance of the polyphenols is great in the brewing process, but the hop suppliers, if you look at their certificate of analysis, they never report the polyphenol concentration in hops. And I would say, and most importantly, what I will show you soon, is the tannoids, you know, on the hops. So even I was chasing on behalf of Nils Oscar, you know, Barjas and Charles Farran, and even Jackie Mashib, to give me information about the batches that we buy in terms of polyphenols. They would say, yeah, we will get back to you, and they never get back to me. And I chase them many times. So why they don't give this information, I don't know, because most suppliers, they do it, and also the analyses are not a big deal to carry out. But it's important that I would suggest, if you guys have the possibility to request this to your suppliers, please do, so you will have a better understanding, because sometimes happen that you have issues of haze and you don't even know why, and then you look at the quality of your malt, and your malt is fine, and then you make sure that your process is running as it should, but and still the haze present, you know. And sometimes you are and then you start being nervous and think you're blaming, you know, the fermentation, the yeast, or any other, or even the water, and it happens that there was a higher levels of polyphenols in the hops, especially when you are heavily dry hop, you know. So that's important. Well, the positive things, I mean, polyphenols has, you know, I would say this dual impact. So it can get really positive effects in beer, but also can give negative ones. So I would say it's such the relevance that they have an impact on the flavor stability, you know, as an antioxidant, or can be also, you know, has an impact on the colloidal stability because of the protein polyphenol complex. So not all the polyphenol is just a name of a huge group of different compounds we're going to go through. So some of them, they're more positive than the others in brewing. Now, the most important ones when it comes to the colloidal stability are two dimers, which are the procyanidin B3, which is basically two dimers because it's a two catechins bound, or prodelphidin B3, which are gallocatechin and catechin together bound. So these ones, they're in the chemists, they call it proanthocyanidins, while the brewers, they know it as just anthocyanogins, okay? And from these anthocyanogins, they are the tannoids, which are basically which I'm gonna tell you soon, the reactive ones to PVP, but I will tell you more in detail about that. Another important thing to know is that winter barley, normally they have higher levels of polyphenols than spring barley. And the reason is, maybe you can associate this because, as we mentioned earlier, the polyphenols, they act as a protection mechanism for the cereal, for the grain, against the environment, of the harsh environment. Obviously, winter also can, protect, the winter varieties they have higher levels. The malt polyphenols are destroyed during the mashing. Yes, as I mentioned earlier, yesterday when you guys define your mashing program, so there is a range of these LOX, which is lipoxygenase, but also there is another enzymes that I mentioned earlier, lipase and the peroxidase and catalase and polyphenol oxidase. All these enzymes, they promote oxidation in the mash, in the wort, and in the mash mainly. So you need to avoid the optimal activity temperature of these enzymes. So and normally all of them, they are in the same range. So I think it's between 55 to 63 degrees Celsius. So that's important. You keep that one in mind. Another thing is the higher the temperatures and the high pH in the mashing, in both in the mash liquor and in the sparge liquor, as we talked yesterday, the more polyphenols you extract. So the higher pH, and this is for chemical reasons, you're going to extract more polyphenols. And obviously the higher the temperatures, that's why some brewers are doing these mash out temperatures at 75, 76 instead of 78. And because they think because there will be a higher extraction of polyphenols. Yeah, it might be, it's a compromise. You might have a more extraction of polyphenols, but at the same time you will you're going to reduce the viscosity, and you will make sure that so there is a complete deactivation of the enzymes with 78. But that's basically the message. Here I just wanted to show you, the different nature of polyphenols. So there are some form from monomers, and they form from monomers dimers to form like catechins and epicatechins, and some others they are formed already from polymer polyphenols. So form big groups of phenol rings, such as the proanthocyanidins, the anthocyanogins, which are the main ones that reacts with the proteins. I will tell you a bit more in detail in the next slides. And tannins, which are high molecular polyphenols. As you can see on the right-hand side, they are really big molecules. So the polyphenols also react as antioxidants because they react, they have, as you can see, many double bonds, and also can react with oxygen, so that's why they can act as antioxidants. But at the same time, as I mentioned yesterday, all big molecules like proteins, like polyphenols, and hop compounds, that we're talking yesterday, oils and resins. They are so big molecules, they help for head retention as well. Okay, so this is one of the positive things of the polyphenols. Now the negative things is that they can be, react with the proteins, and you have problems with the haze. And flavor-wise, you can get astringent flavors, you know, at higher concentrations. Here, I just want to show you all the groups and subgroups of the polyphenols. So, which ones are good? Which ones are actually used as an additives? For example, in AB and other companies, they promote to the brewers to use some tannins at the beginning of the process, you know, or antioxidants, which are combined with metabisulfite together with gallotanins, which has high antioxidative properties. Those are not reactive to the proteins in wort, so they will not participate in the formation of haze. So, and also can work as an antioxidants. On the other hand, if you see the nature like procyanidins or these tannoids, they will be highly reactive to the proteins, which are the bad ones, if you want to call it, for brewing purposes. So yeah. So in short, the message is like not all the polyphenols are bad, you know. So there are some give you some antioxidant properties, and others they will have more reactive to the proteins and problems with colloidal stability. Well, here is in terms of the polyphenol in hops, you know, you can see here the message of this table in the right-hand side is that well, first of all, that there is high, in the left-hand side table, I just wanted to make the point that there's high levels of polyphenols in hops. So it's something that really matters, and we need, as a brewers, we need to look at. And the table on the right-hand side, I just wanted to make the point that there are some hops, depends on the variety, you will have different, obviously the concentration levels will be different. And especially with the proanthocyanidins, the reactive ones to proteins. So you can see here, Tetnanga, they have much higher levels than Hallertau Magnum. And it's not necessarily because one is for bittering purposes and the other one more for aroma purposes. You know, this is something, again, there is not much information by the hop suppliers about these compounds. So it will be good to ask them, you know, so, and then you know, to have better understanding of the colloidal stability of your beer and the oxidation as well.
Andrés, maybe we should take a short break.
Absolutely.
What do you say, 5 minutes?
Absolutely. Oops, I took the wrong. Okay, perfect.
Yeah.
Okay, perfect. So, well, in this slide, which is very informative, as you can see it, this explaining, you know, all these factors that are involved in the beer colloidal stability, and I just marked those which are related to the polyphenols, and as you can see, you know, there's one of them is obviously the concentration of proanthocyanidin in malt, and this in the late 90s, beginning of the 2000s, there were you know, big investments and projects, especially I remember in the VLB 2001, 2002, trying to grow barley, which will be, you know, free of proanthocyanidins. And even I had the you know, opportunity to, you know, to do evaluations between you know, normal malt, you know, barley malt with this type of proanthocyanidin free malt. So, and actually, it was a big Heineken was very interested in this project, you know. So there was one possibility. I don't know how far they went with the project, but they trying to get rid of the reactive, you know, polyphenols in the even from the barley, you know. So so yeah, so this is one of the factors, obviously, the concentration of the polyphenols in the malt, in the anthocyanogens, and specifically tannoids, and the same goes with the hops, which is not pointed out, and because it was back in 2004. This is the source, and they were not very into the dry hopping back then. And another factors is avoiding the use of weak wort. That is something I mentioned yesterday, or the day before. That if you have a very extensive lautering, and especially if you are collecting too much last runnings, you're already extracting on these polyphenols and silicates and everything from the spine grains, from the husk and the spine grains. So that's also will have an impact, so that's why you should not exceed, like as we talked the previous days, to plateau, so if you go below, you are really you want to start extracting some other stuff that you don't want to have in your wort. Also, when you are dealing with high sparge temperatures, because again, you're going to extract more polyphenols, and the use of something to counteract this is using the PVPP, which is well known. I think it's been used since the 60s, if I'm not wrong, to remove tannoids, and this also can be. There is another possibilities even to use it in the brew house. There are some additives that can be a mix of silica gel and PVPP, can be used in the brew house. So you can start, helping, the removal of tannoids during the wort production, and not just in the beer. But here is just talking about the beer. So yeah. Oops. Here, I just wanted to show you all the type of chemical bonds that there are between for the formation of trop. I'm talking about not just hot trop, but also cold trop, and also involved even in the chill haze and the permanent haze. So between the from the polyphenols and the proteins, the famous protein polyphenol complexes, and so they are basically all the bondings present in the nature. They are involved in this. So you can find from hydrogen bonds, hydrogen bridges, and peptide bonds, disulfide bonds, and also there can be ionic bonds between two proteins. But the most important one that just to keep an eye for in this particular case, we're talking about polyphenols, is the hydrophobic bonding. So it's another hydrophobic bonding for this type of polyphenol protein complexes. So basically it's the interaction between the pyrrolidine ring of the amino acid, which is basically the proline, which is the reactive one, and actually is involved in the formation of gluten, and the hydrophobic ring of the polyphenols. So as you can see on the right-hand side on the drawing is, these massive molecules. So the pyrrolidine is this molecule you see here that has this ring of 5 carbons and a nitrogen bound, is reacting with the hydrophobic ring of the polyphenols. So here is when you're dealing with enzymes that we're going to talk tomorrow, there is a different type of proteases that you will hear in the market, but the ones that really helps you to reduce the haze are those who are specific to proline, which is, as you can see, is the one that really reacts with the polyphenols for the formation of protein polyphenol haze. And those ones normally you add it in cold conditions because they are not thermoresistant. Okay. The other protease that you will find, and we talk tomorrow, is going to be more for use it for the breakdown of proteins, more for getting more oligopeptides, amino acids that you want to supply to the yeast, but they're not really related to the improve of colloidal stability of your beer or your wort. That's basically that in this slide. Well, continuing how these proteins and polyphenols react, because it's one of the core things as a brewer you need to know. So how from simple polyphenols they oxidize and they produce polymerized polyphenols, and from there they start they basically become insoluble once when they can warm, when they're warm they can get soluble, but then insoluble when they're cold, but eventually as the oxidation is taking place more and more, and that's what you see in the beer, aged beers, so they become insoluble. Okay, so, and in the right hand side you will see the interaction there between proteins and tannoids, which I'm going to explain in the next slide about what the tannoid means. So how they react to proteins and these specific polyphenols, which are basically oxidized flavonoids, how they start reacting separately, but eventually they create like a rings, like polymers. So that's the way how you start. When you had chill haze, they are in little groups. You have the protein interacting with the flavonoids and when tannoids, and then eventually the oxidized part of the radicals of the tannoids are going to react with another proteins, and they will start forming agglomerates, and then at that point your haze will remain permanent. Here is based on.
Do you mind repeating that one more time? Or is
Which
this
one?
a repetition?
Here, you mean?
Yeah.
So as you can see on the right hand side, you see that drawing, right?
Yeah.
So what you can see is there are simple flavonoids, reacting with the proteins, So based on the reaction that we are talking here, okay? The proline part reacting with the hydrophobic rings, okay? This is what you see here. And if you see there, you can find it, in three fragments of proteins. Well, the proteins reacting with these rings of polyphenols, but the more they get oxidized and they start to be more reactive, the polyphenols side, and then start forming agglomerates, as you can see here. So they start reacting and they start binding all this complex together. So the chill haze normally is when, as you know, the chill haze means that you see haze when the beer is chilled, and when it's warm, it's just you get clear beer. But as the aging is taking place, and the flavonoids are getting oxidized and become tannoids, get more reactive to the proline, to the fractional proline of the proteins or the oligopeptides present in your beer, and eventually it's going to create these agglomerates, and instead of being just a chill haze, will become a permanent haze. Okay, so it's something that you really cannot avoid. It's going to eventually happen, So and that's why it's very important that you need in your beer. You need to keep your all these all these advices, that don't use higher temperatures, trying, especially super important. I think instead of going through a long list of factors, it's very important you keep your pH, as I mentioned in the previous talks, to 5.2. So don't go high, because the higher you go, as we talked earlier, more extraction of polyphenols will be, and that you don't overdo with your lautering, This is another thing. You know that also you have a good quality malt, so you need to double check always the certificate of analysis that you're getting from your malt supplier, and hopefully also you can get from the hop supplier as well, because as you realize, you know, high levels of polyphenols in hops, especially for you guys who like to deal with a high, you know, dry hopped beers, and yeah, and to have a proper mashing program as well, you know, because you have high levels of proteins, which normally you will find a high level of proteins. If you exceed, normally it's about 10 to 11. If you exceed 11%, just to give you an idea, barley, normally you got 12% in raw protein, just barley, pure, not malted. So if you get some malt that you get around the same, that means that the modification was bad, you know. So it's something that you need to discuss with your malt supplier. So just keep those levels of proteins down, and also.
So what level was that again that you said we should keep them under?
Which one? Say again. Can you hear me?
Well, keep the level of protein.
The proteins, yeah. The proteins. When you look at the certificate of analysis of your malt, make sure that that you have no, I will not. It would be incorrect to say low levels. If you have a range between 10 to 11, you're safe. But if you go above that, then you have high levels of proteins, of raw proteins, and that's not good, because also you will have less fermentable extract available in your malt, but also you have problems of colloidal stability. So that's important that you check this in the analysis from the malt. And that's why I know it's not a common practice that homebrewers do that, you know, because they just totally take for granted that the malt supplier deliver great quality malt and the distributor, you know, the online shop. But it's not the case, you know, because we need to keep in mind, it's not because of they're acting on bad faith. It's just simply because you're dealing with a natural product. And of course, it's changing from time to time, from batch to batch. So this is important. So check this always in the certificate of analysis of your proteins. Also check the polyphenols, because also the malt supplier should supply the polyphenols. We're going to talk about the recommended values in the next slide, I think it is. No, in the final slide I have it. What is the recommended values in terms of total polyphenols? What are the recommended values of the total anthocyanogins and tannoids, and in both in wort and in beer, and also the total amount of tannoids in hops? So I will share that in the last slide. You're going to have this presentation, of course, for your own use. I don't know if I could answer your question, by the way. Please let me know.
Yes, you did. Yes.
Okay, great.
And when you said under 12, for the barley values.
Yes. You see, if you find 12% in barley, this is just like it's an average value, of course. But if you see that your malt supplier gives you a barley malt, which is close to 12, when you know that the barley is already 12, that means that they're not doing their job, you know?
Yeah.
Because there is no modification then.
But if that's all we can get right now because of the bad conditions, do we just do a protein rest during the mash?
Yes, this is one thing. You know, if you're following the German purity law, otherwise also you can use enzymes as well. Okay. But yeah, the thing is how you should, you know, give more time to your protease, and normally what you're gonna see in poor modified malt or bad quality malt, you will see higher levels of proteins. As I mentioned in the previous talks, you will have high levels of beta glucans, and you will have low levels of diastatic power. So that's very important. You guys keep that in mind. And when you will get the certificate of analysis, it's like you have all the right to ask to your supplier to give you those figures, because they do it as a routine internally. So they need to give you this values, because normally they provide this to the big breweries, I can tell you. So why then not give it to a small player? So everybody has the right to ask for those information, because otherwise you can have a big issues in your process. So when you see that, which is normally what you're gonna see now, and especially, for example, we see this like for organic products, because of normally organic raw materials normally have poor in quality, you know, and so because they don't use, just because the way how they were grown, you know. So and so what you need to do is you need to prioritize, or to, you know, to put more attention to your protease rest, and the same goes, but normally it's all together. You need also to extend your beta amylase rest as well, you know, and if just to make sure, and also you're going to have big issues of beta glucanase, you know, the filterability of your mash. So if you are not married with the German purity law, what I suggest you to not have nightmares is to use enzymes, exogenous enzymes or commercial enzymes. There are some enzymes that they have already the entire, which we're going to talk tomorrow. You have protease, you have beta amylase, sorry, alpha amylase, and you have and hemicellulases, which means beta glucanases, polulanases, and helanases, which is all these hemicellulases that you find in the mud. All this stuff, they're products already like a cocktails. But now, if you only want to focus on filterability, also there are just glucanases, pure glucanases. But it's advisable, you know. So because now those people who are dealing with German purity law, they might struggle a lot to have a good brewhouse yield. Another way to do it is that you can also do decoction method, you know. Also that helps you to improve the attenuation of your beer, you know. But also it's not guaranteed that you will have problems of filterability, you know, because you have too high levels of beta glucans. So all this goes together. I sound like I'm going off the subject, but all this is related, you know. Higher protein levels, you know, higher levels of beta glucans, less fermentable sugars available, and also low levels of the diastatic power or enzymatic activity of your grain, you know. Okay.
Great, thank you.
Yeah, no, my pleasure. Here, just wanted to show you, know, that still the different type of polyphenols. You have the simple ones, so the preformed polyphenol complexes, you know, how they oxidize and then they become active polyphenols, and they react, you know, with phenol nitrogen complex, and they make trop and haze, you know. So it's just it's a general knowledge, you know. So all this happens simultaneously. Here, that's a nice table, and this is something also you will hear. And I think I already mentioned this when we were talking about brewing considerations. There is the mash filter and the lauter tune, as we know. So with the mash filters, as we know, we need to reduce the particle size, you know, the finest we can do, you know, like in the lab when you're measuring the fine extract. So that's why they're using hammer mills for mash filters, while for the lauter tune you're using all these mills that we were talking, you know, 2, 4, 6 roll mills and conditioning mills and so on. So the thing is that Germans, brewers, they claim that they don't like to use mash filters, but I think it's more for commercial market reasons, because they say that there is a major extraction of everything, which means of beta glucans and polyphenols, everything what is in the grain, which in theory is the answer is yes, but again, brewing is not like a black and white thing. Also depends a lot on the contact time, okay? Because the mash filtration with mash filters is much, the residential time of the grain on the mash filter is much shorter than you have the spent grains in your lauter tune, okay? So it's like a very intense but quick. So so Miura, which is as we know, the Belgian company who create these mash filters originally, and doing a lot of trials and trying to prove that wrong. But here, we are talking about polyphenols, and what you can see here is that there is an increase of polyphenols between the lauter tune and a mash filter, but it's not really big, okay? So this is important to you know, and by the way, the polyphenols, the maximum, according to the MEVAC or DBC, recommended values should be no more than 200 milligrams per liter or ppms, okay? So that these two, they are in the upper limit, but they're still within range. So, and if you compare the other figures, you know, there is not really a difference for quality purposes. So that's the only point I want to make, that there is not really a negative impact using a mash filter. But you need to, as I was mentioning in the previous talks, you need just to be careful in some specific features, like we were mentioned, like avoid the shear forces. You know, it needs to be a super gentle mashing, because, you know, the more you reduce the particle size, obviously the more beta glucans might be present, and that will have problems with the filterability. So shear forces is important to keep it to minimum when using mash filters, and oxidation as well. So that's why as I mentioned, all what the Miura develops in terms of brewing technology is based as a core, their mash filter, and from there goes all the things around. So that's why they're trying to, you know, to sell to the brewers, you know, milling systems with that you are adding CO2 to, you know, strip off oxygen. The same goes with the pre-mashers and have a very gentle mashing, and, you know, all that stuff, in order to have the best conditions to run the mash filter. Okay, and these ones are so quite interesting. So you will see the gliadin is the protein, the major protein found in malt, wort to be precise, and the gliadin is what contains the proline fraction, which is the one we were talking that is reacting with the polyphenols. And on the other hand, you have the tannic acid, you know, which is the tannins which react with this proline. So here, what you can see, the message on these diagrams is that, well, the relation, you know, of the concentration of tannic acid and gliadin in relation to the formation of haze. Now, the haze is measured normally in NTUs, which is nephelometric turbidity units, but also can be measured in EBC units, and also can be measured in OD units, which means optical density. All of them, they are basically on spectrophotometry. The only difference between them is the way how they pass through the light, in which angle they pass through the light, through the sample, you know. But normally, when it comes to turbidity, the most common one, I would say, is the NTUs, and EBC obviously for brewers, but normally it's an NTUs, and also you can measure this. Well, specific instruments, but also there might be the possibility to do it with the spectrophotometers. And the OD is like a measuring a single wave. I think is at the final rank is at 600 nanometers in the spectrophotometer, and also they use it not just for turbidity, but also they use it for yeast counting. So this is one message. Well, the message on the diagram on the left-hand side. On the right-hand side, which is more interesting, is the relation between the formation of haze, the range of pH, and the alcohol content. So what you can see here is that the information that here telling you is that the lower the alcohol content, you know, in a range between 3 to 4, you know, the higher the turbidity. And the same goes with alcohol. When the alcohol reached to start increasing, let's say between 10 to 12, also at pH between 3 to 4, also there will be an increase of haze. Okay, so that's an important information, you know. And yeah, what else? Oh, here. So this is the important thing. This is very important. You don't need to learn by heart, of course, the structure of the molecules, but this is the main proline fraction I've been coming from the proteins, which it will make a polyproline. I mean, it's gonna make a polymer of it, and that one gonna react with the PBPP or with the polyphenol, so the tannoid. Now, what is the difference? What is a tannoid? You know, and here you can even read it. It was Chapon, this brewing scientist in the early 90s, report that the tannoids are present in beer between 10 to 60 milligrams per liter. And tannoids are those polyphenols that can react with PVP, okay? So you need to be clear, it's not PVPP, but PVP. And you can see in the footer what's the difference between them. In any case, both are polymers, which is like just picture, if I can say it in rough words, it's like a fake proteins, you know, but it's dependent also on the charge. So basically they're going to interact with the polyphenols, and that's why the classic additives as a clarifiers or as a for beer, you are using silica gel or silica sol, which are silicates that this gonna pretty much have the same. They're gonna mimic basically a polyphenol, so it's gonna react with proteins. So all silica-based clarifiers are gonna react with proteins, while all those PVPP-based additives is gonna react with polyphenols. So that's why brewers sometimes combine both, and then they attack from both sides. So from the reacting with polyphenols with the PVPP, and also reacting with the silica-based products with reacting with proteins, just in order to clarify your beer. Now, as you know, the most common among craft brewers, they normally use silica sol and adding this in their beer. You know, more you know, prioritizing the clarification from the protein side, but also they also consider using the PVPP. Now, so basically tannoids are, as we know, the most reactive ones. They're from the chemical theory is the anthocyanogens. That's why brewers, they analyze this. But from these anthocyanogens, they are the reactive ones. The real most reactive ones are the tannoids. And this guy Chapon basically wanted to basically name those polyphenols that react with this PVP, and to say, well, these are the most reactive polyphenols which are involved in the colloidal stability of the beer. So that's the way how brewers in the lab, you know, they measure the tannoids. So we'll have a better understanding about the reactive polyphenols in the beer. Now, this is a very important slide, and I think it's the last one. Yeah. So I got this information from the MEVAC. As I mentioned, is the most important, you know, I would say the most strict guidelines. They are more strict than the EBC. They are based on the EBC, but they are like more detailed. And these are this ruled by this German Brewers Elite, and the EBC is, of course, the European Brewing Convention. So I just here collected, you know, the specifications and recommended values or normal values. So you can see here, the normal value in beer goes from 150 to 200 milligrams per liter of total polyphenols, but those not necessarily all of them react with proteins. Okay. And then there is the other type, type of analysis, which is the anthocyanogens, which they react with hydrochloric acid, and thus the normal value is between 50 to 70 milligrams. So you can see, you can make your your relation between, you know, what is from the total polyphenols, which ones are the in terms of concentration, which ones are the reactive ones. And from there, you have the tannoids, which is the one that Chapon is proposing. And then here's when you compare, you know, how much you got in terms of the malt, you know, how much you got in terms of the beer, and how much you got in terms of the hops. And then you will say why you got more in the malt than in barley, you know, because during the modification, during the malting itself, there is also, there is more formation of anthocyanogens. So, but not in terms of polyphenols. Polyphenols remain the same. Well, not remain the same, but it's linear. I mean, there is no increase of production of polyphenols. But the important here that you guys need to keep an eye is in terms of in the beer, how much tannin you have in the polyphen in the beer, and also to compare, you know, which this is one of the, so I've been just repeating myself, but it's interesting to look at. It's also the tannoids present in hops. So again, I would love that, you know, hop suppliers, they give in their certificate of analysis, not just giving you the alpha acids and the hop oils present, but also can give information of the tannoids present in the hops. And yeah, that's it. I don't know if you have any questions.
I know you mentioned something about the antiseptic and antibacterial
Yes.
things in polyphenols.
That's right.
Is it a huge benefit from those polyphenols if we talk about hops?
Yes.
Or is it just a small?
Yeah, no, actually, that's as we know, and that's why the famous story of the IPAs, you know, that they were increasing the levels up in, you know, those times to export it to India, you know, from it's a natural preservative. So yes, yeah, it works, but I don't have really information about what is the ideal concentration of polyphenols to act as a proper antiseptic. But as you know, guys, when those who are into sour beers, they know that there are some bacteria that they cannot, you know, they cannot deal with in the presence of, you know, of hops, you know, and that's why the brewers who are dealing with sour beers, especially those who are into bacteria and stuff, they're trying to keep it the bitterness or the presence of hops to the minimum to not affect the performance of their microflora. So yeah, that's true. But I do not have information in terms of concentrations and stuff. But yeah, it's an antiseptical, if we can call it that way. Yeah.
Yeah, because you hear a lot of, if you think about lambic brewers, they always talk about the beta acids from hops.
That's right.
But they never really
they don't
mention
talk about polyphenols. Yeah.
No.
Yeah, but it's also can work on. Yeah, but the problem is, as we know, the polyphenols, we have the problem about the colloidal stability, you know, and also if you can overdo, you can also have problems of astringency. And actually, you can also extract polyphenols from the wood, you know, and that's why, you know, get this astringency when, or as we call woody flavors, you know, overpowering when you're dealing with oak chips, for example, or barrels. So there also you can extract a lot of polyphenols. I mean, there is a huge levels of polyphenols in the bark of the trees, you know. So that's why we cannot eat bark ourselves, you know. But yeah, but that's an interesting, for sure. It has an effect, but I cannot tell you the concentration. But it's we should look it up, you know, in the literature.
Yeah, thank you.
No, My pleasure.
I'm sorry. I'm still just trying to understand the last page that you went over here. You said it was the most important page, and I'm just looking at it and trying. How should I?
It's not the most important, but it's important, guys, to keep in mind in terms of the concentrations, and also to be familiar with it, you know, because it's something that just normally malt suppliers and hop suppliers don't provide this to the brewers because they don't ask for it, you know. But they can give you a better understanding, you know, especially when I know that it's one very common question from brewers that they are facing problems with haze stability, you know. And sometimes they try to find, you know, all the ways possible, and they don't understand why still have this haze, and it's my dry hops, and it's the beer, it got contaminated. And it could happen because of, you know, the hops, you know, the dry hops, you know, the polyphenols of the dry hops can have my affecting this, you know. And also if they're oxidized as well, you know, because as I told you guys, the polyphenols are very, they're antioxidants. So all like the antioxidants, you know, like ascorbic acids and polyphenols and this, the way how they work, just in a very general way, they capture the oxygen present in the medium. So if you exceed the concentration of these antioxidants, and then instead of being antioxidants, they become pro-oxidants because they start capturing oxygen all over the place, you know. So this is, sorry, I probably may go off from your question, but that's one of the reasons why I say that it's important that you get more information from your suppliers in terms of polyphenols and tannoids, you know, anthocyanogens. Yeah. When I say anthocyanogens and tannoids, not everybody do the tannoid test. Normally in the UK, they do more tannoids, you know, and while in Central Europe, they perform more anthocyanogens. I don't know, maybe for philosophical reasons, I guess. But yeah, but both are common. Yeah.
Okay, so these are tests that you can perform.
Correct.
Okay.
Yeah, not you. I mean, unless you have you have a lab set up in your brewery. But these three parameters, total polyphenols, anthocyanogens, and tannoids, they can be performed by the malt suppliers, labs, and the same goes with the hop suppliers, you know.
Okay.
Yeah, might be a very unusual question for the hop suppliers to ask for this, but it makes totally sense to know it, you know, especially when they know that brewers these days, many they use, you know, high levels of hops in dry hopping and late additions, you know, because it's major, you know, we're talking about a lot of hops. So and nobody talk about the concentration of polyphenols by the hop. They always talking about the malt, as we know. So that's another new subjects to look into it. But all these three, you can, the malt suppliers, they perfectly can do it for you, you know, because as I mentioned earlier, they normally do a much more extensive list of analysis internally, you know, especially when they're, you know, buying barley from the growers, you know, they need to make sure that they meet the standards to be able to malt high quality malt, you know. So you brewers, you have the right to ask for it, you know.
Okay.
Yeah, that's what I'm doing. When and in Oscar, there are some figures. Sometimes they send me some very poor certificate analysis. Say, well, yeah, thanks. That sounds just like a routine or just like a formality, but it doesn't say nothing to me. You know, I need more information just to make decisions. You know, how to adjust my mashing programs or what I will expect. You know, because otherwise, will be once you make the beer, it's just nearly impossible to know what happened. You know, before that. You know.
So how are you going to react to one of these? If you get one of these tests, say the anthocyanogenic test.
Anthocyanogens, yes. If I have it,
if it's too high or too low.
Yeah, well, then you need to, in that case, you need to blend. You need to talk to your malt supplier. Say, well, look, you're telling me they need to give you, better quality. But if you already process the wort and then you're trying to blend it with another batch, which has not such a high levels of polyphenols, or otherwise, you depends on the beer style, otherwise you will have really problems of, of haze. And even if you filter your beer, you will have problems of chill haze,
Yep.
because they're so tiny compounds. It seems like a big molecules, but those go through the filters, even if you are dealing with stereofiltration, they go through, yeah, and they start reacting with the proteins. So that's something that you cannot deal much, because in the case of the proteins, yeah, you can work with enzymes, as I mentioned earlier, that can break down the proline. So this is one solution, So I say, okay, I know there's high levels of polyphenols. I can treat my beer using a protease in the cold beer, like this brewer Clarex, which is basically a protease that is has affinity for proline fraction, which also is using and at the same time you can produce gluten-free beer, because the proline is one of the is involved in the formation of gluten, So that's the way to go. If you're already too late, if you have already beer and you didn't check the polyphenol levels, but if you know it before you start brewing, and then you know what to do. Yeah. I don't know if I answered your question.
Yes, you did. Thank you very much.
No, my pleasure. Any other question? Maybe we finished today earlier. You can ask me questions from the previous talks if you have any as well, of course.